Hi there, it's Ryan Darcy again. Congratulations, if you're watching this module, you have gotten through Module 1, Brain Basics, and we're moving on to Module 2, which is really to focus on the specific results of neurocatch and train you to be able to understand the waveforms. And today's module is about the same length as Module 1. I'm just going to do a quick review of Module 1 just to refresh you, and then we'll dive into Module 2. So in Module 1, we did the basic brain components that contribute to neurocatch. Specifically, we talked about neurons and the fact that the neurons in your cerebral cortex that are involved in specific functional areas generate the signals we record in neurocatch. We talked about the fact that when you break down into the major parts of a neuron, it's the input side of the neuron at the dendrites with these postsynaptic potentials, either excitatory or inhibitory postsynaptic potentials that create the electric potentials that get recorded by EEG sensors of the scalp. We talked about the fact that the raw EEG you can then pair with ways to stimulate the brain and specifically extract the smaller signal embedded in that of how your brain is processing a stimulus. So whether or not it's a tone or any other sort of stimulus. And then we talked a little bit about how that is really strong in the temporal measures, so in millisecond resolution, and we can get some spatial information, but the neurocatch data should always be seen in a larger sort of ecosystem of neuroimaging technologies where we can use measures like magnetoencephalography or functional MRI if we need to further understand how brain function works. So in today's module, we're going to dive specifically into how neurocatch works and kind of view it as popping the hood and looking underneath. So to start, we want to first think about this whole concept of stimulating the brain. So how do we stimulate the brain in neurocatch? Well, the first thing is that one of the key things of event-related potentials is that they've existed in labs for many, many years, almost coming up, I say, since the 1930s. So the implementation of stimulating the brain became clinically accessible in neurocatch because we came up with a specific sequence that compressed our ability to get these responses to make them short and rapid. And here what you can see in the top part of this graph is what we did is we started by using basic auditory tones. So you put in earphones and you hear these basic auditory tones that get you ready for hearing a word pair that either matches or doesn't match. So what we can do is we can repeat this sequence multiple times to get lots and lots of stimulus examples and put that into the signal averaging. And in neurocatch, the goal is to be around 5 or 6 minutes. So we get as much information in a short possible time. Now, how does this work in terms of the tones? And let's just focus in on the tones. So in the literature, there's a very well-established experimental study and stimulation method called the oddball paradigm. And what it does is it gives you an array of standard tones. So these are set, you hear them, they don't change. And every once in a while, at a sort of an unpredictable and rare and infrequent time, when you don't expect it, it'll give you a change in that tone. So you can imagine, for example, that you would put a louder tone in, which is what neurocatch does. So that becomes called a deviant. That's the oddball, right? And from an evolutionary standpoint, this should be fairly intuitive in the sense that you could be walking in the forest and all the sounds are standard and you expect them. And then if a stick snaps, it sort of is a deviant sound that you would pay attention to. And that's exactly what the neurocatch does. So we're going to talk about an auditory sensory response just to processing the loudness of these tones called the N100. And then we're also going to talk about processing this deviant tone, this unexpected attention. So you have to use basic attention to say, okay, what was that? Which is this P300. And then for neurocatch, the N100 and the P300 come from the tones. And if we move over to the words, that's where we get the cognitive processing response called the N400. And here what we do is we basically use a key, a simple word pair, and specifically word pair primes. So for example, if I say hive, your brain likes to automatically load up the association with bee. And if I say sled, your brain will load up the association with snow. And so these would be what you'd call matches or congruent word pairs. And what we can do is in NeuroCatch, we vary that with mismatches. So when I say hive, instead of giving you something that you would expect, like bee, I give you something you don't expect, like snow, which would be an incongruent match. And these incongruent matches elicit this N400 response as you're cognitively processing that this semantic relationship didn't match. So what's kind of the magic of NeuroCatch is that this was a separate experiment in research labs. This was a separate experiment in research labs. And what we did is we managed to take that into one sequence that instead of running in 3 hours, runs in an automated sequence in 5 minutes. What do you hear? You basically hear these tones, 5 tones, that cue you to get ready to hear these word pairs so that you evaluate whether or not it matches or mismatches. And you hear that a number of times over the duration of 6 minutes so that we can basically probe your brain for these known responses. So now what I want to do is start with the first of many pictures for you to get familiar with what these responses look like. So we're really training you to be a pattern recognizer. The human brain is the best pattern recognizer there is. We still haven't created artificial intelligence and computers that can rival us. So this module is really about getting used to seeing these responses in their raw brainwave format. Now, NeuroCatch will give it in radar plots, and I'll show you those later and make it easier. But for boot camp training, what we want to do is get you to start to be used to seeing the features in these waveforms. So we take them from looking like squiggly lines on a page to I can spot these responses. So let's start first with the top left-hand corner. And what you can see here is this is the N100 response. So what does that mean? It means that the way this is named is the N is in reference to the polarity of the peak. So if it's a negative going peak, it's called N. And if it occurs around 100 milliseconds, then it gets the name 100. So N100 stands for a negative peak around 100 milliseconds. Not exactly at, but in the vicinity of. Now, you might notice this is a little counterintuitive because you would have expected that the negative peak would be down and not up. We can credit electrical engineers in the dawn of time for this convention. And what you will notice is that in the NeuroCatch report, we've gone and flipped it around to make it innovative. But in the papers, in the scientific papers, it is by convention that N gets plotted up and positive gets plotted down. So of course, now that you know how to name the N100, we can go and we can look at this P300. So this is a positive going peak that occurs around 300 milliseconds. And you can see this peak right here. And finally, the third one, which we'll talk about, is this negative going peak around 400 milliseconds. And this is to the word pairs, the ones that in red don't match. So this is a negative going peak around 400 milliseconds. Your brain's saying, hang on, that word pair didn't fit. So now what I'm showing you is I'm showing you the results for adults, healthy adults for age 20 to 30. And what you're seeing is on the x-axis is time, right, in milliseconds. This is the 0 start point. So this is the point at which the tones were presented, right? Every tone started at this time. And this is the microvolt axis. So we're looking here at how big is the response. So we know now it's a negative going response. You can see that this N100 is about -4 microvolts. This P300 is about 8 microvolts. And you can also see that the responses to the tones tend to be a little bit larger than the responses to the words and the cognitive processing. So you see the scale has changed from -6 to 6, where this is -8 to 12. So these responses tend to be a bit bigger. Now let's take and walk through this healthy adult sample. This is an average of a group. So you're getting kind of a composite look at what it looks like. And there's a couple things that you need to train your eye for. So the first thing is, if we go to our 0 point and we go back, this is recording before the stimulus. We call this the pre-stimulus baseline. So what we would expect here, Remember, I was teaching you about signal averaging, right? If there's random electrical activity, it'll average to 0. Well, here is that in practice, right? So we have not stimulated the brain at this time, and what you can see is the line is flat. So it's averaged to near 0 because there's just random electrical activity. We're not actually picking up anything specific to the stimulus because the stimulus hasn't been presented yet. Now, once the stimulus is presented, the clock starts, so to speak, right? So in the early time, this is, well, the tone has hit your ear, and within 100 milliseconds, it goes from your ear to your primary auditory cortex. So it takes only 100 milliseconds on average for most people to process a tone. Some people will process them faster. Sometimes they will be later. I'll show you later on that if you have a hearing impairment, you would expect the timing for this to change. Now, what's interesting is if you look at these two colors, you can also get a sense that the deviant in this case is the louder tone, right? And the blue is the standard. That's the tone that's the standard level. And then we increase the loudness, that's the deviant. So if you look, what you can see is that the louder tone has a little peak right before this N100. We call that a P100. I know it's confusing because it's at the same time, but we actually call this the P1, and then this becomes the N1. And you can see that the size of this response is bigger because we played a louder tone. So our sensory response, if you increase the sensory input, you get a larger N100. And you can see that if you compare that to the tones that are softer, you still get the N100, but it's a smaller peak. So what's important here is this is a very standard waveform shape for your brain processing sensory information. It's called the P1, N1, and P2, P3 complex. And you see the P3 here. I'll show you and tell you more about the P2 later on. But this response is something we see quite commonly in our sensory processing at the early stages of sensory processing. And what's really interesting is if you look, when you move from tones to looking at sensory words, so these are now words, this is speech you're hearing, you see the same thing. So let's go through that. The baseline is flat because there's no stimulation. Then you hear the word, you get this early positive waveform at around 100 milliseconds. Then you get an N100, and then you get this sort of P2, P3 component, and then the N400. So notice this is a very characteristic sensory response of your brain processing sensory information. Now, let's look at them again, but let's change the age. So let's go look at adults, healthy adults aged 50 to 85, and go through and let's do exactly the same thing. So first of all, our baseline is flat. These are tones. And then at the time the tone onsets, you see this early positive peak, this P100. Then you see this N100, and it's bigger for the louder tones than for the tones that are not so loud. And that goes into this P300. This is that peak that is that unexpected, right? This deviant tone. It was like a stick snapping in the woods. Your basic attention to the difference, and you're processing now. You're processing at a sensory level. This is you processing attention going, hang on, that wasn't right. That didn't fit. That was a rare, louder tone. And then you can see they return back. And this all happens in the space of seconds, right? This is 300 milliseconds. Now, when we look at, okay, those tones got you ready for these word pairs, and these word pairs are either going to match or they're not going to match. So what we see is before the word pairs, it's flat, everything looks good. Around 100 milliseconds, there's this sensory processing, the P1, there's the N1, then it goes down into the P3. And then for these mismatching pairs, we get this very smaller but very significant peak that's processing the cognitive difference, this N400. Notice that you see this both in our, no matter what, if you're a young adult or an old adult, you see this pattern, you see how characteristic and how replicable, how this pattern replicates. But you can also see actually that it changes a little bit. So look at the P300 timing for your older sample versus your younger sample. So it's also tracking how fast you process information. And we do, of course, know that as you get older, you process information a little slower. It's too bad because I've got too much gray hair, but it does happen. This is showing how sensitive neurocatch is in detecting that. And it's showing that in both cases, you get this N400, but you're showing an early processing in younger age that's more sensitive to this. And we're going to go into this peak, and I'm going to tell you a little bit more about this. But what you can tell here is that unlike a tone, which is discrete, speech actually comes out over time. And so you can see that this peak is much broader and as your brain processing over time, whereas this peak and this peak are much sharper because it's a discrete tone. So these waveforms are a good representation of your brain processing information in millisecond resolution. So let's go to the next page. It's going to look identical. The fact is, if we do this in 100 people, we get this result in 99 or 99.9. So when we go from a group, which you're just looking at a group picture, to a single individual, this is what it looks like in a single individual. This could be one of your patients, for example. And again, let's walk through it. Flat baseline on the tones. You got this large N100, which is larger in the deviance, the louder tones. It gives this P300 in terms of attentional processing, that was a deviant and your basic attention is kicking in. You see that whether or not you're a young adult or an older adult with a little bit of a delay in the latencies. You can see the latencies are a little later, but you see the same pattern. And when we go over and we look at how are we processing these word pairs, flat baseline, you get your sensory response in both cases, and then you get this N400 response. Notice this 2 peak. This is actually breaking down that speech information. So when you hear something unexpected, the first thing you hear is an unexpected sound, and that's this peak right here. And then you start to process that it didn't fit the meaning either, and that's this peak right here. Notice that does change a little bit in this particular example of a young adult versus an old adult, and that is an important thing to pay attention to, and we'll get into that a little bit later. All right, so now you've seen kind of the main waveforms, you've got familiar with that. Let's take a look at how some of these things change. So let's take a look at the N1 and P300 in terms of what you're used to. So this is the top line. So this is, if we present this with earphones in the auditory modality. So again, flat baseline, there's your N100, there's your P300 for tones. See it? Now what I'm showing you here is something a bit different. I'm showing you for the 3 sensors. You'll recall from module 1, I taught you about FZ, CZ, and PZ. So these are sensors on the front, center, and parietal lobe position. And you can see that this response changes across these sensors. If you look at the N100, it's much larger, and is the P300 in the FZ and CZ sensors in this particular case. And it tends to fall off towards the back. What's interesting about this slide is you can now compare, what if we did this and we didn't present it through hearing, but we presented it through visual. And so the bottom line shows you the same experiment in visual. So again, flat, there's your N100 right here, and then there's your P300. Notice that this looks different. And what you see is this earlier component, it's a positive going peak around 200 millisecond called a P200. So again, you see these responses. It doesn't matter if you get them in the auditory modality or in the visual modality, but they do change on the way that they came into your brain. And this would make sense just if you think about in the next slide, we'll show you speech versus reading. And in reading, when you read a word, it's instantaneous. In speech, it takes time. So let's take a look at the next slide to see how that shakes out in terms of this record of how your brain processed it. So here it is. This is the N400. And let's walk through again, flat baseline. We get an early sensory N1, P1, N1 peak, and you can see that as you have before. And then this large extended peak. There's your N400 with this double asterisk. Now remember I told you that there's an earlier peak that's to the sound of speech. That's this phonological, what's called a phonological mismatch negativity. So early on, as early as 250 milliseconds, your brain is starting to code. That word didn't match and the sound didn't match. And then later on it's saying, yeah, that's right, the meaning didn't match too. And there's a lot of research that's unpacked this to understand how our brains process language, and they're highly reliable characteristic features. But the important part for this module is let your inner experimentalist compare to when I hear speech to when I see an instantaneous image of a word. And look at the shape of the peak for the N400. Again, we see the flat line, we see the sensory perceptual waveform, But then notice that we see a much sharper peak because there's not the information being processed in speech over time, but rather we can access it much more rapidly. And so the N400, when we present it visually, is a fundamentally different peak. And to drive that point home on this slide, we don't see any speech-related activity. We just see one peak rather than multiple peaks embedded in the same waveform. And you can see that here. All right, so now you've seen the N100, the P300, and the N400 in a group comparing to young and old, and individuals showing it's the same in young and old. And now you've compared it in terms of whether or not you hear it or you see it. What about where does it come from in the brain? Let's step through that. So let's take them one at a time. Let's talk about the N100 first. So I've told you previously that that's the time it takes from your ear to your brain, right? And your cortex. And in our brain, in our early on in module 1, I showed you that in the temporal lobe and the posterior temporal lobe is where our primary auditory cortex is. And this is just some experimental results that show if we stimulate your left ear only, so our auditory system, like many systems in our brain, are crossed. So what happens is we see that when we look at the activation map of the N100 result, it shows that it's actually asymmetric and that it shows activity greater in the right ear because our systems are crossed. So stimulate the left ear, you're going to get activity in the right ear. When we then look at that in terms of MEG, so we say, okay, let's find out where that came from in the brain. Let's use MEG, do this experiment. It localizes to your primary auditory cortex. And you can see that localizing here. So in the case of the N100, it is a low-level sensory process that you would expect in terms of information processing from your ear to your brain. And if you have auditory pathology, you might see that change, particularly in latency. Right? And here's the spatial underpinnings of where the N100 would be localized for if, for example, we only stimulated your left ear. Well, what about the P300? Well, remember in module 1, I taught you that brain function can be localized, but it can also be part of a larger distributed system. And as we go up in information processing, as we go from lower level to more complex levels, we start to use more distributed networks in our brain. So this is just showing you on the left. This is a bit of a busy graph, so I'll walk you through it. This is the sensors at FZ, CZ, and PZ. And this is showing you basically the sensory processing and the P300 and making the point that there are actually different types in the P300 family. So I want you to pay attention to this P200. This is a positive going peak at 200 milliseconds. This is what's called a P3A, so it's a variant of the P300. This is another variant called the P3B. This is kind of the classic P300 you see. So this is the one we're looking at in terms of a target, this red one. So this is the neurocatch one. And in this experiment, what they can show is that when you look at the P300, it has a broad distribution across the scalp. So you can see all this red activity is showing you the peak of the P300. And when you look at different variants, this P3A versus this P3B, all of a sudden you can notice that while they're distributed generally in the same area, the distribution, the shape of the peak on your scalp is different. And this is much more of a sharp peak in terms of your scalp. Now, what we can do is we can actually go and look at where in the brain that came from. And so on the right-hand side, we're just looking at the activation maps. This is for this P3B. And the important point to realize here, this is your color code activation. So basically, if you see reds or greens, this is the location of the P300 generator. And what you can tell is it's very distributed. You get, this is an auditory P300 and you get auditory activity, but you get parietal activity, you get frontal activity, you get occipital activity. The P300, because it's attention, it uses a distributed network in the brain. So where the N100 was much more localized, you would expect, because you're going up in information processing, you're now starting to use much more distributed neural systems. So what about the N400? Well, the N400 creates a similar sort of thing. Now note that it's a negative going peak, so we expect to see more blue here. And you can see this is a broadly distributed central distribution on your scalp map. And this is actually on the right-hand side is the neurocatch N400 in MEG. So what you can see here is we know in general that language tends to be, particularly for right-handers, tends to be left hemisphere organized, so it's stronger in the left hemisphere. And you can see the classic language areas using not only the temporal lobe, the primary auditory and the inferior temporal lobe, but also what we would call sort of the posterior temporal and the interface with the parietal lobe. So this is Wernicke's area for those who know that sort of concept. And you can see that it's also extending into the frontal lobe, into Broca's area for expression, and the dorsolateral prefrontal cortex. So an extremely classical language presentation for neuroimaging for the N400. This is cognitively processing the word pairs. You can see that that is not just in your left hemisphere, it's also in your right, activating much of the same homolog areas, but to a lesser extent. Now, of course, the sanity check is if we flip and we say, all right, I don't want to know what the activity for incongruent greater than congruent, show me the words that matched, you see no activity. So what we're seeing with the neurocatch is really these violations of what you expect, localizing to your language centers of your brain, again in a distributed network of how you are cognitively processing. All right, so now some clinical examples to give you a feel for how this would look clinically. So in the N100, this is quite an interesting study to give you a feel for this. I'll break it down for you. What this was a study that looked at normal hearing level people in both a quiet and a noise environment, and then individuals with auditory neuropathy, again in a quiet and a noise environment. And what they did, you can see it on this horizontal axis, is they actually varied the intensity level. So the sound of the tones went from being quite high to lower in intensity. And so what we can do is on the left side, let's just look at the latency of the N100 component. What the peak timing, what happened? So in our healthy individuals in a quiet room, you can see that as you increase noise, the latency speeds up. So when you go from a low, like a quiet environment to you can definitely hear the tone, it optimizes around 100 milliseconds. So a fast N100 is around 100 milliseconds. And if you got into a quieter environment, it goes to slows and is a little slower at about 140 milliseconds. Now, if you add noise, background noise, you can see that even in healthy individuals with no hearing problems, that has a sharp effect. So as you add noise, you slow the latency response down. And as you get into a louder and louder environment that overcomes that noise, or tones that overcome that noise, you can catch back up to optimal processing. Now, when you compare that to your auditory neuropathy, you see that in our quiet environment, the people with auditory neuropathy look a little worse than even our healthy individuals in a noisy room. And when we put them in a noisy environment, then they look quite much worse. And their response is slow to as much as 200 milliseconds. So this gives you a good normative range in terms of would I expect to see ideally an N100 or 100 milliseconds. If I start to see an N100's latency starting to get into the 200 millisecond, I would certainly think that that was in a delayed range. Now on the right-hand side, amplitude, pretty much the same thing. As we increase the loudness of the tone, we get a larger N100, and you can see there's a little figure showing you the response size. This is for your healthy individuals in a quiet environment. When we add noise, we see that that's still, we can still return to an ideal amplitude size, but noise does make that worse and reduces the amplitude. You can see it gets smaller in terms of the negative scale. And that this, again, matches with auditory neuropathy in a quiet environment. And then when we put people with auditory neuropathy into a noisy environment, they get much worse. So that gives you a sense of how, from an auditory sensation point of view, the N100 maps this. Now let's take a look at the P300. I apologize for these graphs. This was the best resolution we could get from extracting. They're very important graphs, so it's important to show. But let me walk you through it. Again, on the left is the latency. On the right is amplitude. And on the horizontal axis is age from early years. So 0 is at the far left and 100 is there. And you can see that this is a large sample of P300 data across the lifespan. Each one of these dots corresponds to the latency for an individual. So this line shows you your age-related trends for P300 latency. And we can see is early on in life, we may be a little slower with our P300s in our developing brain. This is at 400 milliseconds here. And as we get to our 20s, we're in our peak area. So when we're in our early 20s, we have our fastest P300s. And then as we age and we get older, they start to delay over time. You can also get a sense of the relative dispersion of this, so that you can see that from about sort of 300 milliseconds to 500 milliseconds is the typical window that you might see the variance in this P300, and that it is quite variable when you're young and then stabilizes into adulthood. It's a very similar function for amplitude. So if we go over to the right and we're looking at amplitude, again, you can see that when we're young, we have a smaller P300, and then our larger P300 comes around our 20s. And then as we age, our P300 amplitude reduces. And you can see that there's a fair amount of variation when we're young. And there actually, in the amplitude, does maintain a fair amount of variation across the lifespan. And it's somewhere between, let's say, 2 microvolts and 20 microvolts, or 40 up to this area. So the P300 can be quite a large response, and it is very much focused on how we process information, that basic attention, processing information. Clinically important if somebody's had a bad sleep, or if they are drinking coffee before or after. We know these factors affect the P300. This is really based on how's your information processing today, and how's your attentional processing at the time of testing. So if you want to optimize a P300, you would make sure people are awake and alert and aware and in their best possible case, and you would see those effects much more in the P300, whereas an N100 would be a basic sensory processing related to hearing and that sort of thing. All right, last but definitely not least is the N400. Again, I'm showing you a comparison of how the N400 latency on the left and amplitude on the right changes as a function of age. Now this is a little more restricted. For latencies, we're showing from age roughly the 20s to 80s. And then for amplitudes, I'm just showing you into older years from ages 50 to 80. And what you can see here is the basic story holds true, that our latency for our N400 delays as a function of age. And it delays roughly in your 20s, it's somewhere from 350 to 450. And then as you get older, that goes to about 400 to 500. So we would normally expect N400s to be anywhere in the 300 to 500 range. And if you're younger, you would look a little earlier and expect some faster latencies. And if you're older, you wouldn't be surprised to see them past 400 milliseconds. Amplitude, a similar sort of relationship. Remember, again, this is a negative response, so you want the response to be down. So when you're in your early 50s, you might have a larger N400, and then as you age, your N400 amplitude will go down. In general, it will go down to about 2 microvolts. A response that's not 2 microvolts or 2 microvolts or smaller, I would flag. So basically, for any of these components, I would look around 2 microvolts as your minimum response. And the N400 in general is a smaller response than the P300 and the N100. So you don't expect it to be that much larger than 6 microvolts, so 2 to 6 microvolts. Again, amplitude largely allows us to know if the response is there or not. It can change depending on a number of factors. So the big amplitude isn't necessarily good. If you have auditory sensitivity, for example, you can have a large N100, and as you treat auditory sensitivity, you can see that amplitude reduce. Timing, however, is a very absolute metric of how long that response took in terms of how long your brain processed information. All right, so let's put it all together. For the end of Module 2, I just want to take you through some clinical examples using concussion. So this is work that we did in ice hockey players for concussion testing. We'll start on the left-hand side. The basic experimental design was to use NeuroCatch to measure them at baseline in blue. If they got a concussion within the first 24 hours of that concussion, and then when they were cleared clinically to return to play. Now, the result looks confusing, but this is a good graph for training, looking at ERPs. Let's go back to the beginning of the module. First of all, we're looking for the flat baseline before the stimulus starts. Then we're seeing that there's this P1, N1 going into a P3, so the sensory attentional peak to the tones. We are seeing in particular that the amplitude increased as a function in the early stages. So if you compare the blue to the red, you can see the increase in the amplitude and the delay in the latency. So in early acute concussion, because your brain has undergone a concussion, likely you're using more cortex to do the same response, hence the larger amplitude, but it's taking you longer, hence the delay in latency. And you can see that effect is quite consistent across not only the N100, but also the P300 and the N400 down below. So here's the N400, and you're seeing that the N400 at baseline with a larger amplitude and a delay in latency. Now, on the right-hand side, these are athletes that didn't have concussion, where you compare their responses after. These are junior A hockey players, a lot of hitting, a lot of checking, and a lot of subconcussive suspected injury. And when we compare their baseline to their end of season, we don't see a difference in this N100. So you see no difference in this N100, no difference in the P300, but you do see a difference in their cognitive processing. Now, here's a great opportunity to showcase where we're going with NeuroCatch. You're in Electrophysiological Bootcamp to get familiar with these waveforms. But our goal in NeuroCatch is to give you clinical results that are really much easier to read. And so you can go and look at the waveforms underneath. But for ease of use, what we've done is we've taken these results and we've put them onto a radar plot. And this radar plot gives you a fast and easy way to look at the results without having to dig into the specifics that we've done in this module. You'll have them there for reference, you'll have them there for comfort, but when you're looking at the NeuroCatch results, this is the same data presented on a radar plot. So we have three responses, right? The auditory sensation, both its amplitude and its latency, basic attention, both its amplitude and its latency, and cognitive processing, its amplitude and latency. So 6 total. And when we look at these athletes at their baseline, we get a hexagon that's quite symmetric. This radar plot is scaled in percentiles. So it's sitting around the 50th percentile and showing you a healthy group of athletes prior to a season. When we take a look at the concussed athletes, and you saw that the amplitudes got larger and the latencies got delayed, look at what that does to the result. The amplitudes getting larger increases the result, and the latencies getting delayed reduces this radar plot result. So the shape shifts from a healthy hexagon to a triangular format, which is a pretty impressive. We're pretty excited. We're looking in the science and research if this can be used as an electrophysiological biomarker of concussion, which would mean, by the way, you wouldn't necessarily need a baseline, and you could monitor your athlete who came into you with a concussion to get them back to a healthy hexagon level, which is a standard that you can use. Now, when they were returned to play, this is just to drive home the point that they start to go back towards a healthy hexagon shape. You can see this is starting to look a lot more like baseline, except NeuroCatch was specific and sensitive enough to detect that even though they were cleared to clinically return to play, there was still a significant difference in their basic attention metric. And when we looked not just at the athletes with concussion, but the athletes with the subconcussive, we saw a significant difference in their cognitive processing latency. And the research has since gone on to show this and show differences between ice hockey players in Bantam and Junior A, and also just recently hot off the presses, similar replication of differences in football and other sports. So this is kind of the clinical value of NeuroCatch for what we know it to be today in our research, is that it allows you that important objective cognitive function measurement to benchmark and or to track treatment. And you can now not just know, hopefully, when you look at these waveform results, what you're looking at in terms of these 3 measures, but you can also start to map what they mean when we show them to you on this radar plot. And with that, we're at the end of the second module. The third module is to go through some clinical examples, yours and ours. And congratulations on both the basic brain bootcamp and underlying reading NeuroCatch waveforms. We will stop recording here.